All right, let's go ahead and get started with our last class of the quarter. Announcements. Pre-final will open tonight. I know you've been looking forward to this. So I hope you all have on your calendar when you are going to sit down and take this. When it closes on Saturday, it's closed for good. There's no remake of taking this up again because the next day, the IAs are going to hold a review session to go over the pre-final. We have set it to be podcast. Hopefully that will happen, but hopefully a lot of you will also come, be here in person with the IAs going over all of those questions. Just a reminder to please submit these set forms. I think they might be due this Saturday even, so get some extra credit for the class. We will, in addition to this review session, we will also have some exam week office hours. So if you have last minute questions, Tessa will have a 2-hour period on Tuesday, and Hamangi and I will also have sessions on Thursday before the actual final. So you have multiple opportunities to come in person, get everything clarified. If you don't have time, or if you need an answer to a question pretty fast, put it on the discussion board. We are looking at that every day, and if there's questions there, we can answer it, and then it's available for the whole class, because if you have a question about something, probably other students have that same question. And this, of course, is in preparation for the final, which will be here in this class next week, starting at 3, and we have a 3-hour period to take this. Any questions? All right, just a reminder, do not cheat. All right, this is especially true for the pre-final, right? This is an unproctored online exam that I give to you so you can practice and figure out what you need to really focus on to study for the final. Remember, you only need to get half of the questions correct to get full credit for the pre-final. So take that opportunity to study and take it as if it were the real thing. Question? Did you not upload these slides? Are they uploaded? These slides will be uploaded immediately after this class. Every single slide I'm showing tonight. And the reason for that is I was working on them up into the last minute, even incorporated some things based on my office hour today. So I think this is a good opportunity to really sit back, just pay attention. This will be podcast. If you want to go take notes once you have the notes posted, that would be a good opportunity. We're going to have a lot of clicker questions, but everything will be available to you as soon as I can get back to my office and post this. One last thing I want to say about this, do not cheat. So we had an issue a couple years ago where some students got an email, said, hey, let's work on the pre-final together. We can do the questions together. And this made students very nervous because they thought they were going to be accused of cheating. If you receive an email like that, report it. You can report it immediately to UCSD. Let me know about it. Let the Office of Academic Integrity. Turned out that the person sending that email was not even in this class. I don't know what they were trying to do. But don't get nervous if something like that happens to you. Reach out. If you have questions about am I allowed to do this or I'm not, just ask. You will not be penalized for asking any questions about what you can or cannot do. So, given that, any questions? No? Good. All right, so let's briefly review what we talked about in class on Tuesday. So the main theme of that class was the molecular biology of cancer. And what we learned is that cancer, tumor formation, is really just your normal cells picking up mutations and then starting to divide out of control. And so normally that's going to take multiple mutations in order to overcome all the regulation that normally goes into making sure your cells only divide when they are supposed to divide. Only when the proper signals are there to, we need to heal this wound, so we need to have some cell division. The types of mutations that will take a normal cell to a cancer cell are going to involve the cells needing to sustain proliferative signaling. In other words, they need to think that they need to keep dividing, and there's various ways to do that. They need to evade growth suppressors. So there's a lot of negative feedback that comes into cells that says don't divide, and so they need to get beyond that. The cells are going to need to enable replicative immortality. What does that mean? What do they turn on usually? Telomerase, right. Replicative means replication. If you're going to be doing DNA replication, you're going to have that end problem all the time unless you maintain your telomeres. And so cancer cells very often figure out a way to turn telomerase activity back on. If they can't do that, they're not going to be very successful at dividing. Their DNA is going to decay away, and you're not going to end up with a tumor. There's also going to be, the cells are going to recognize this is abnormal, maybe we shouldn't be dividing, maybe we should just kill ourselves, go through apoptosis, so the tumor cells are going to have to resist these apoptotic signals. They're going to have to evade the immune system. Your immune system is always surveying when it sees strange things happening, like cells dividing in an area where it shouldn't. Very often, your immune system will take care of that problem. So a successful tumor will need to evade the immune system. Once the tumor really starts to grow, it often will induce angiogenesis. What does that mean? Yeah, new blood vessel formation. Why does a tumor want new blood vessel formation near it? Bring in the nutrients, bring in the oxygen. We need all this so that we can keep dividing. And then also by generating these new blood vessels, that allows the tumor to metastasize. Metastasis is when cells of the tumor break off, enter the bloodstream, travel around the body, and then can potentially take up residence in other organs, other places in the body, and that's when it becomes very, very difficult to treat the cancer once it has spread. All right, so for all these cellular changes to happen, there need to be genetic changes. There need to be mutations in our genes. And we talked about two basic types of mutations that can lead to cancer. We can have gain of oncogene expression or activity. So these are going to be genes that can promote cell division, that cause proliferation. Normally, they are tightly regulated. These genes are tightly regulated. They're normal genes. They're proto-oncogenes. But once there's a mutation that causes them to be overexpressed or take on a new activity that is going to support proliferation, that's when we consider it to be an oncogene. For example, we talked about RAS. So RAS is this GTP binding protein. When it's bound to GTP, that's its active form, and it's going to promote cell division. RAS itself is an enzyme. It is a GTPase, meaning it has the ability to hydrolyze the GTP. When it hydrolyzes the GTP, it's no longer in the active form. If you have a single nucleotide change that could change the amino acid that's needed for that GTPase activity, you're going to get RAS stuck in the GTP form. That would be an example of how the activity of RAS has changed, but not necessarily the expression level. You might have the same level of RAS protein, but now it's always the active form of the protein. We also talked about a transcription factor. This MYC transcription factor that turns on a lot of genes that promote cell proliferation. And there's a variety of different ways of how you could get too much MYC protein so that it starts to activate genes that should be actually silenced. On the flip side, we have tumor suppressor genes, and it's going to be the loss of tumor suppressor expression or activity. Again, either transcriptionally downregulating that tumor suppressor or picking up a mutation in it so it no longer can be an active transcription factor, for example, p53. For these types of mutations, for a proto-oncogene to become oncogenic, do you need both alleles to be mutated or just one? Just one, right? It is a gain of function mutation. The other one's wild type, still regulated, no problem. But if you have a gain of function or overexpression of the other allele, that already puts you into oncogenic activity. What about tumor suppressors? Do you need mutation in one or both? Both, exactly. So The last thing that we talked about are microRNAs and their roles potentially in cancer. And we said whether a microRNA is going to act as an oncogene or a tumor suppressor depends on its target. It depends on its targets in that cell type. For example, RAS, one of these signaling proteins that can become oncogenic if it's overexpressed, is repressed. It has target sites to the LET7 microRNA. So if you're a tumor cell, you want to get rid of LET7 microRNA because then what's going to happen? No more regulation of RAS, high levels of RAS, you promote cell division. So in this example, the LET7 would be acting as a tumor suppressor to keep this growth-promoting gene under control, regulated. P53, this is that tumor suppressor that's lost in a lot of different types of tumors. It actually has target sites for miR-125. Would it be loss of miR-125 or overexpression of miR-125 that would lead to oncogenesis? Overexpression, exactly. You have suddenly very high levels of miR-125. It's going to reduce the expression of P53, similar to losing P53 activity. Does that make sense? All right, so let's do a clicker question. So which of the following are correct? Madeline?
Like somebody that like smokes or like is around like cancerous material or something like that, like carcinogens, would that like target the like factor or the DNA itself that like can create cancer?
So for it to create cancer, it means when the cell divides, it needs to pass on that trait. So you need to change the DNA.
Okay, so it's not just like the daughter cells; it has to be the DNA itself.
The DNA would pick up the mutation, and then when it divides, that mutation would be copied into the DNA that goes into the daughter cell.
Okay.
So carcinogens mutate DNA.
Okay, all right. Thank you.
Ten more seconds. There's only 106 of you here today. All right, final answers. All right, E, all of the above. Can it be all of the above? Let's see. All right, who wants to share their thinking? Nick.
You have predisposition for cancer. You'll have a loss of function in one of the mutations.
Yeah, so A is basically the definition of predisposition. So predisposition to a cancer happens when a person is born with a loss of function mutation in one copy of P53, which is a type of tumor suppressor. Exactly. So predisposition has to do with tumor suppressors. You already have a loss of function in one copy. The odds of you losing that activity by getting a loss of function mutation in the other allele are increased. So A is definitely correct. What about B? Proto-oncogenes promote cell proliferation. Is that true? Yeah, that is their normal activity. What about oncogenes? Do they promote cell proliferation? Absolutely. And what is the difference? Regulation, control, exactly. Proto-oncogenes are still tightly regulated. When they get overexpressed or a mutation that allows them to not be regulated, that's when they then convert to an oncogene. So B and C are correct. D, homozygous loss of the P53 tumor suppressor can cause insensitivity to therapies that stimulate tumor cell apoptosis. Well, it must be correct, right? Because we're looking at all of the above, right? So what does that mean? How is P53 related to apoptosis? Yes? Oh, we'll go with you. Yeah, so P53 is a transcription factor, regulates a lot of genes. Some of the time what it'll first do is say, wait, stop, let's not go into the cell cycle, let's not do any replication. Then it'll turn on repair genes, so you can repair any mutations. Too many mutations, things seem terribly wrong, it will turn on the apoptosis pathway. So P53 activity is needed for apoptosis in many different cell types. If the tumor cell has already lost P53, then certain types of chemotherapy that rely on apoptosis of the tumor cells and rely on P53 signaling to allow that to happen are not going to work in those patients. They will be insensitive. So yes, the answer was all of the above. Any questions about that? All right, so thank you, all of you who sent me all of your suggestions for what we could possibly review tonight. I apologize, we definitely do not have time to go over every suggestion that I got. So what I am going to go through are what I received the most suggestions for. And if there are things that we don't go over tonight that you would ask me about, please put it on the discussion board, come to office hours, go to discussion sections tomorrow. There's lots of time to still get clarity on anything that is still confusing. So what we're going to do is we are going to review the list of different experiments that you have learned in this class. It is important for you to know the basic steps of all these different experiments so you know why you would do it and what you can interpret from it. And we'll use chromatin immunoprecipitation or the CHIP experiment as an example. We'll then talk about directional cloning, why you would do this, how you would design your insert with restriction sites to put it in the correct orientation, and then how you would use directional cloning to make reporters and how you would interpret data from reporter experiments. And then finally, we'll also review CRISPR. All right, so here is the list of all the different experiments that we have talked about in class. Hopefully this all looks familiar to you. And so as I said, it's important to know the basic steps that we talked about in class of how you would do each experiment. Students in the past have told me that they had a lot of help by making note cards of these experiments and actually like writing down the steps and then drawing out a figure of what the data might look like so that they can think about what am I actually doing this experiment for and what am I looking at. And so when we're talking about understanding the steps and what you can learn from an experiment, we will go through chromatin immunoprecipitation. So hopefully this looks familiar to you. And so if you are going to do a CHIP experiment, what is the very first step you would need to do? You'd have your cells, and what'd you do? You cross-link, yes. And so we use chemicals, sometimes UV radiation. And what that does is it causes covalent bonds between DNA and closely associated or bound proteins. All right, so you have your cells, they're all cross-linked, you isolate the nuclei, and then what do? You have huge chromosomes that are all cross-linked together with proteins. Yeah, break it up, right? Yeah, so you break it up into smaller pieces. Sometimes we use mechanical force like sonication. Other times we use enzymes, so these long cross-linked pieces get broken up into smaller pieces. These smaller pieces will still have the DNA that's blue wrapped around, in this case, nucleosomes, still covalently attached to whatever proteins it was associated with in those cells. What is the next step? What are we going to do? This is where the IP part comes in. So we have chromatin, we broke up chromatin. We're going to use an antibody to immunoprecipitate based on whatever protein we're asking about. If we want to know, is our gene associated with TBP, we would use an antibody to TBP. In this example here, they're using an antibody against the acetylated histone, let's say histone 3, has acetylated on lysine 9. So every time the antibody sees that protein, it is going to stick to it. The DNA is still covalently bound to it. everything else gets washed away. Everything that does not have this protein that the antibody is designed against is going to be removed. So this green, these are the acetylated nucleosomes. They will be immunoprecipitated or selected. Immunoprecipitate just means we've used an antibody to select something. So we have selected these chromatin chunks that have acetylated histone 3 here. And then the last step we're going to do is we want to analyze the DNA. So we release or we purify the DNA. And so all of the DNA sequences that were associated with this histone are going to be in this pool. Any DNA sequences that were not associated with that protein gets washed away. They are not in your template pool anymore. And what is the final step you can do to then detect, was my gene associated with this protein? PCR, exactly. We do PCR. So here are the steps exactly as they were previously in your notes. And at this last step, we're using PCR to determine if a specific gene or DNA sequence was associated, because this last step here, when we release the DNA, this is going to be all the DNA. So what if I wanted to know all of the DNA sequences that were associated with my acetylated histone 3 lysine 9? What would I do instead of PCR? What could I do with this pool of DNA? Instead of PCR, I would sequence it. Exactly. Remember, we can use next generation or this parallel sequencing to determine all of the sequences in one experiment that were associated with this protein of interest. All right, so those are the steps. But why would we do this experiment? Why would we do a chip experiment? What do we want to learn from this? What's the point of doing a chip experiment? Lydia? Yeah, you want to know the specific gene or all the genes, all the DNA that is associated with a protein of interest in a particular cell type, a cell undergoing differentiation, under infection conditions. They're always going to be in vivo situations. Question? You can get an idea if it's more heterochromatic or more euchromatic. And so, for example, if you're looking for the acetylated histone, what that's associated with, is that going to be more associated with euchromatin or heterochromatin? Euchromatin, exactly. But there's a better experiment if you want to generally ask if something's euchromatic or heterochromatic. What is that experiment? DNase protection, exactly. All right, so exactly as Lydia said, so we use this experiment if we want to identify factors or proteins associated with a specific DNA sequence in cells under different conditions, different cell types, in the presence or absence of a hormone or a drug, etc. Here's some questions. So does CHIP show direct binding of a factor to DNA? No. What experiment do I need to do if I want to see if this protein binds to this sequence of DNA? You could do footprinting. What else? An EMSA. Both of those questions, both of those experiments will answer that question. Do I see a footprint? Do I see a space where the protein was protecting the DNA from DNase and footprinting? Do I see a shift because the protein bound to my piece of DNA and couldn't migrate through the gel as quickly? Does CHIP show if a gene is expressed? Does not directly show, but can it give you information about whether a gene is likely expressed? Yeah, exactly. So, as you guys answered, so CHIP does not show direct binding because things that are closely associated with the DNA can still get cross-linked. We talked about this example where this activating protein GCN4 is directly bound to this UAS sequence in the DNA. So it would give a positive CHIP signal. It would also be positive in footprinting or EMSA. But it recruits co-activators like this GCN5. So if you did a CHIP, GCN5 would show up that it's associated with this gene. But if you threw GCN5 in a test tube with this sequence of DNA, you wouldn't see a footprint and you would not see a gel shift. This question here about whether the gene is expressed. So we can make predictions about expression. What protein would we want to use an antibody against to get a really good idea if a gene is being transcribed or not in a chip assay? TBP, yeah. So if you have TBP or any RNA polymerase II or any of those general transcription factors at the gene, then it's most likely going to be transcribed. If you wanted to directly ask if a gene is being transcribed, if RNA is being made, what experiment would you do? A northern blot, yeah, you would look for the RNA. All right, so here's a clicker question about chips. So to analyze the association of specific proteins with the DMPK promoter in cells at different stages of differentiation, you perform a chip using antibodies to these proteins based on these results. And which cell type is TBP most likely associated with this promoter? Ten more seconds. Final answers. All right, C. Why do you like C as the answer? What clues did you use to come to that answer? Khalil. Did you choose C? I guess. Mm-hmm.
Like, you check, so it's the only one that's acetylated, so that's most likely means it got spread,
Exactly. So when you're thinking about gene expression and TBP and RNA polymerase coming into a promoter, that promoter needs to be open. It needs to be available. The acetylated form of histone H3 is the open euchromatic form. If it's methylated and closed up, it's going to be very hard for those general transcription factors like TBP to get in here. And so based on all the evidence you have here, this is the only cell type that has a lot of acetylated, and therefore the myotubes is the answer. Any questions about that? Yes? Let's go on to the next slide for a second. It's going to be used in some other questions. So, so that's the answer. All right, so let's go through looking a little bit more in depth at the data we have here and thinking about some other types of experiments as well. So when you're looking at this type of chip experiment and you see a band, what actually is that band? What does the band represent? Is it protein? It's DNA from what? What's the last step of your chip? PCR. So you've done PCR to specifically look if the DMPK promoter sequence was associated with these proteins. So the band is the DNA that got amplified with your primers. Is that clear to everyone? All right. All right, so now I do have this NHR stands for nuclear hormone receptor. I called it M. So based on the data you see here, which type of nuclear hormone receptor would you guess that NHRM is? Remember, there's two general types that are regulated differently. So is it always associated with the DNA? No, we don't see it there in the undifferentiated or myoblasts. So which type of nuclear hormone receptor is only associated with the DNA in the presence of its hormone? The homodimer, exactly. Remember, the homodimers have O's in them. They're stuck out in the cytoplasm. Heterodimers are the ones that are always bound to their DNA binding element. And depending on whether the hormone is there or not, they can act as a repressor, recruit co-repressors when there's no hormone, recruit co-activators when there is a hormone. Because we don't see it under all cell type conditions here, the best guess here would be that this is acting as a homodimer. And then what does association of each protein suggest about activity of the gene? So if you take into account everything you're looking at here and what you know about these states of the histones, how is the activity of this gene regulated? What would you say? Is it on and undifferentiated? Probably not. It's pretty highly methylated chromatin there. Methylation starts to go down, and then you have acetylated once you're in myotubes. So it is probably repressed and undifferentiated, and during differentiation starts to come on and finally is expressed in the myotubes. All right, some more questions. How would you test if differentiation affects the interaction of these proteins? So this is differentiation. These are the same proteins with another gene. What would you change in your experiment? So we're looking at DMPK here. You want to look at another gene. What would you change? Antibodies. You want to look at the same proteins but associated with a different DNA sequence. Primers, exactly, primers. Or do next generation sequencing and get all the sequences at once, right? What if you wanted to test if HP1, so a different protein, interacts with this DMPK promoter under these same conditions? Then what would you change? The antibodies. You'd use an antibody for HP1. What if you want to know if these proteins associate with the DMPK promoter in cardiomyocytes? What would you change in this experiment? You'd change the cell type. You'd do it in cardiomyocytes instead of these cell types. What if you wanted to know if the mRNA is expressed in each of these three different cell types? Northern blot. You collect RNA from each one of those cell types. You do a northern blot. What about protein? Is the protein expressed? Western blot. What about if you want to know if the chromatin structure associated with this gene is more condensed in myoblasts compared to myotubes? Which experiment? DNase protection, right? Is it open and therefore chopped up by DNase, or is it tightly compact and therefore protected? And finally, what would you do if you wanted to learn if this promoter has negative regulatory elements? Mutagenesis, you make a reporter gene with that promoter hooked up to GFP or something, and then you could first do deletion analysis, but then you'd eventually want to do mutagenesis to really pinpoint those regulatory elements. Any questions about that? I'm thinking about an experiment. All right, so then the idea of reporters leads us into our next topic, and that is directional cloning. Why would you do directional cloning? So directional cloning means putting an insert into a vector in one orientation, right? An orientation you want. So why would you want to do that? Yes? Go. So you want it in the exact spot and you want it in a certain direction or orientation. So what would you be making? Why would it be important to have orientation? Yes? Yeah, if you wanted to express a protein, it's very important that the start codon is near the beginning of transcription and it's not flipped around. So if you want to express a protein, and what's the other thing you might be using, or using recombinant DNA to make an orientation will be very important? Yes?
A vector or an insert, right?
So the vector is what we're putting the insert in, and the insert is going in the correct orientation. So if you want to express a gene or a reporter, exactly, a reporter gene. So directional cloning means that we want the orientation in a particular manner. It is important the direction that the insert goes into. This would be important in reporters, and we'll go through some examples of how we would build a reporter and why it's important to have your insert in the correct orientation. And then also expression constructs. If you want to program bacteria using their promoter to express, for example, insulin in the bacteria cell, it's really important what orientation that open reading frame goes in front of that promoter. So that transcription will start in the correct orientation. You'll have the start codon at the beginning and the stop codon at the end. All right, so let's talk about reporters. So we use reporters to basically identify regulatory sequences. The first time we talked about reporters, we talked about using them to identify regulatory sequences in promoters. But then later in class, we considered the idea that any regulatory elements could be tested for regulation by hooking them up to a promoter, hooking them up to a reporter gene. And so the basic idea behind a reporter is that you're going to use a test sequence, maybe a promoter, maybe a UTR, and you're going to fuse it to something that's easy to detect. I think we almost always use GFP as the example here. So when this protein, if GFP is expressed, you shine fluorescence on it, you see bright green. And then you're going to compare the expression of your reporter with the intact test sequence to those where you've systematically mutated versions. So maybe you've systematically mutated sequences in the promoter to find activator or repressor sequences. Or maybe you've made a reporter with a 3' UTR and you're going to mutate sequences in the 3' UTR to see where you get changes in expression of your reporter. Because when the expression is different from the unmutagenized, that means you found a regulatory element. All right, so let's go through an example of how you would create a reporter gene using directional cloning. So suppose we wanted to find or analyze this 3' UTR of your gene of interest for potential regulation. Here is our vector, which already has a promoter that will drive expression in the cells you're putting it in. Here is GFP coding sequence, so it will produce a GFP messenger RNA, which will be translated in the cells to GFP protein. And you want to use this vector to test whether there's regulatory sequences in the 3' UTR. First thing you need to think about is, where am I going to clone this UTR into my vector? Where would you put it? After the GFP coding sequence. You would put it exactly in the same position as where it normally would be in a gene. What if I were analyzing the 5' UTR? Where would you put it? Yeah, you would put it before the GFP coding sequence. You would put it upstream of GFP coding, but you would definitely have the promoter in front of it. So wherever the sequences normally are that you're studying, that's where you want them to end up in your reporter gene. All right, so suppose I give you this sequence. I tell you here's a variety of different restriction enzyme sites that are available in this reporter. And we already established we want to put the 3' UTR at the end of this GFP coding sequence. Which enzymes would you use? Would we use this? No. This one? No. This one? No. So that already rules out Hindi3. Would we use both EchoR1 and BAMH1? Yes. That is how we're going to get it to be directional. We are going to open up our vector with both of those enzymes, and then we're going to put those same restriction sequences on either side of our 3' UTR. Is it important for this 3' UTR to go in the correct orientation after the GFP coding sequence? Yes. Remember, there's going to be polyadenylation signals here. If you put it in the wrong orientation, those signals are going to be on the other side. They're not even going to work at all. So you want the 3' UTR of this gene to be in the same orientation in your reporter. And this is where we're going to use different restriction enzymes in order to allow that to happen. EchoR1 is closest to the stop codon here, right? The way it's drawn. So we want that to be at this position of our insert that we're going to make. And then BAMH1 we would put at the end. By putting these restriction enzyme sites on the correct size, we're going to make an insert that will only fit into our vector in one orientation. Those sticky ends, once we use the restriction enzyme, are going to let those puzzle pieces only fit together in one direction. Does everyone see the idea behind this and what you want to do? Right. So we already have our restriction sites inside our vector. But now if we want to clone this UTR sequence into this vector, what do we need to do? How are we going to get this insert so we can put it into the vector, clone it into the vector? What method? Yes, Madeline. Absolutely, yes. So we're going to use PCR to add these specific restriction sites onto either side of this 3' UTR. And so you'll have an opportunity to design that with this clicker question. So which of the below primers would you use for directional cloning of the 3' UTR downstream, this is downstream, of the GFP sequence in this vector? Yes. Oh, what? I even have HDMI today.
I'm sure you'll be glad when you're done with this class.
Yes. This then. All right. Okay. So weird.
It's a $1 billion machine.
Oh, I'm showing you the answer, huh? Sorry about that. All right. Well, let's see what you guys put for your answer. Maybe some of you changed it now, huh? Yeah, most of you are putting A and anyways, and hopefully you got there the correct way, but we will go through it and make sure we all understand how to do that. So, go ahead. Oh, wow, this is, okay. All right. Okay. So, the answer indeed was A. And so I'll take you through the steps in case you don't see how to do that. Most of you got it correct, so I think you're clear on this. But what you want to do is you want to insert the restriction enzyme sites on the 5' ends of your primers. Remember, the 3' ends need to perfectly base pair, and then on the 5' ends, you can add any sequence you want, including the restriction enzyme sites, so that you will have Echo R1 at this position, and then BAMH1 on the other side. And so the way you would design that is the way we always design our primers. We make sure that the 3' ends are going to base pair to the template. So this was the sequence I gave you. I always think it's useful to write it as a double strand. You only need to draw it for the 2 ends. Imagine it denaturing, and then draw how your primers will be able to base pair to that DNA template so that the 3' ends will face each other. Synthesis, remember, is always going to be from the 3' hydroxyl, going in that direction, going in that direction. And so in your first round of PCR, you will have this synthesis, and then what you'll end up is from this 1 double strand of DNA, you'll now have 2 double strands of DNA, and 2 of those pieces will have the initial beginning of that restriction enzyme sequence. These products, after this first round of PCR, then become templates for the next round of PCR. So these products will undergo denaturation. So here's the products. We denature them. The same primers are in your tube. They are going to anneal. They're going to base pair, and you're going to get synthesis again. Now you have 4 strands of DNA, and what you can see is you're starting to copy that restriction enzyme sequence because the primers become part of the DNA products, and the DNA products are the templates for the next rounds of PCR. So the end result is going to be many copies of your insert flanked by the restriction enzyme sites that you wanted. Does anyone have any questions about designing these primers? You will definitely have to design primers, probably with restriction sites in your exams. So make sure you're clear about this. Yes, question.
Why don't we put the restriction enzymes right after the promoter instead of after the GFP?
All right, so that comes to here. So the insert is 3' UTR, 3' untranslated region. So where is that located in reference in a gene? It's right after the coding sequence, right? So there's a stop codon, translation stops. Everything after it is 3' to it. It is untranslated, so it's 3' UTR. So if you're studying a 3' UTR, you want to put it in the same position in your reporter as it is in the natural gene. Does that make sense? So this is a 3' UTR, so we are putting it after this GFP coding sequence. If it were a promoter, we would put it upstream of the coding sequence because that's where the promoter would be. Question?
Professor, can you go back to the original picture? For the question.
This?
No, for the. Yeah. In here, like, we only have eco-RI in the fields. Yeah, we don't have it anywhere else. Would it also work if you only, like, design it like one restriction design?
So if you only put one restriction enzyme on either site and only cut this with one restriction enzyme, what would happen to that insert? It could go in the right way 50% of the time. It would go in the wrong way 50% of the time. You would not get directional cloning. The reason we use 2 different enzymes is so that the pieces can only fit in one orientation, which you'll see in a moment here. All right, so we have our insert. We cut our insert with the restriction enzymes. Yes?
Sorry. Okay.
That's okay.
With the 3' end, though, it has to match base pair, and then the 5', so upstream, we don't really care, right? It could just be.
When you're designing a primer, the 3' end has to base pair perfectly to the template. The 5' end of the primer can be the restriction enzyme sequence.
Okay, which would just be the same as the template, right?
The 5' end you'd want to be a restriction enzyme sequence. That's the new sequence you're putting on. The 3' needs to be able to base pair to the template. Okay. All right, so you cut the insert, and then you use those same enzymes to cut your vector here. And so what's going to happen when you cut, so the little piece of DNA that was between EchoR1 and BAMH1, you just wash that away. And now look what happens. You have an overhang of EchoR1 from your insert that's going to fit very nicely into this overhang site that was created by cutting the vector with EchoR1. These sticky ends, or like pieces of a puzzle, are what allow the insert to go in a particular orientation. If you flip this around, it's not going to be, you're not going to have base pairing. You're not going to have orientation. And so this is why using two different enzymes are going to allow these pieces to fit nicely together. And when you mix them together, add ligase, you're going to reestablish those same restriction sites on either side, right? So the end result is you now have a reporter that has that test 3' UTR after the coding sequence where it should be. Does everyone understand that? All right, so let's say this 3' UTR, we wanted to test if there's a sequence in this 3' UTR that is regulated by LET7 microRNA. How would you design an experiment to test if this new reporter you've made is regulated by LET7 microRNA? Would you expect expression to be different in the presence or absence of LET7 microRNA? Yeah, so you would put it into, sorry. This says LET7 microRNA underneath this. This is the GFP expression, and this means target site. So you would take your reporter, and you would put it in cells that either have the LET7 microRNA, so that's the control, and you get a certain level of expression. Oh, no. So, but what's weird is I'm doing both HDMI and the UBC, and they both seem to be. Oh, okay. Don't do anything. Okay. All right. So then you test it in cells that you don't have the LET7 microRNA. Maybe you've used an oligo to inhibit its activity. And then you look, is there a difference in expression? Did we see a difference in expression? Yeah. So what happened when LET7 wasn't there? Expression went up. We would interpret this as LET7 is negatively regulating the expression of our reporter. So less expression in the plus LET7 means that we're getting repression. The next thing that would be important to do is, all right, is this really through regulation of this site that I think is in the 3' UTR that LET7 targets? So now what do I want to do? You would mutate the site, exactly. So now you make one version that has the target site and another version. So this version has the target site, this version does not. What happens when you remove the target site and LET7 even is still there? Expression goes up. You can have all the microRNA you want. If you don't have the target site, it's not going to be able to recognize it. It's not going to be able to regulate it. It's this very similar idea as when we were making mutations in promoter sequences and looking for regulatory activity. Any questions about this? All right, so the final topic we'll go over tonight to review is CRISPR. So remember, CRISPR was actually discovered as a natural antiviral defense in bacteria. It's a way that bacteria are able to store information in their genome about phage that had previously infected them, and then to use a system that involves a guide RNA and an endonuclease to recognize that phage DNA and degrade it anytime it sees it again. By understanding that we can target an endonuclease, a DNA endonuclease, to any sequence of DNA just by having it be complementary to a guide RNA, This is, of course, very powerful and has allowed us to engineer editing of DNA in just about any cell type. So if we're going to set up a CRISPR experiment, there's a few things you'll need. You will need the guide RNA. The guide RNA is going to determine where you want to make the edit in the genome. That guide RNA is going to need an endonuclease protein. Cas9 is the one that we've talked about in class. There's many different types of CRISPR proteins that do this. But the protein's going to need the guide RNA to take it to a specific position in the DNA genome and cause cleavage. So you get double-stranded cleavage through the target DNA. Once you have a double-stranded break, there's two options for the cell to repair this double-stranded break. One option is through non-homologous end joining. So remember, during this mechanism, you're always going to get chewing back, nuclease activity. Sometimes some random nucleotides get added. It's mutagenic. That's why we call it error-prone. And that might be fine in your CRISPR experiment. Maybe you just want to design a series of mutations. And so all you need in that experiment is your guide RNA to target the specific gene you want to mutate and the endonuclease, Cas9. There are other times where you might want a specific mutation or a specific insertion of sequence to happen. In that case, you're going to need to provide one more ingredient in your CRISPR experiment. You're going to need to provide the homologous DNA sequence. Sometimes we call that a repair template. It is the template DNA that you want homologous recombination to use in order to fix that double-stranded break. The key for that is the homologous repair template needs to have homologous or the same sequence that flanks the break you made. And then between that sequence, you can put any sequence you want. You could put GFP sequence if you wanted to insert a big sequence. You can make a single nucleotide change to repair a mutation. Anything you can think of, basically, you can design to have in your repair template in order to have it incorporated into the edited DNA. All right, so here's an example of CRISPR-edited worms that we made in the lab. What we did is, so worms have argonaute. Argonaute, you know, is essential for the microRNA pathway, so we wanted to study the expression of this argonaute. We used CRISPR to add a GFP tag to the endogenous gene. So how would you design a CRISPR experiment to tag your favorite endogenous gene? What would you do? What would you need first? You need the guide RNA, right? And how would you design the guide RNA? Complementary or the same sequence, right? Because we're double-stranded DNA. It's going to base pair to either side, right? So it needs to be specific for the gene you want to edit. So we have the guide RNA. Sometimes we call it an sgRNA for small guide RNA. Yeah, we need the endonuclease, so the Cas9 endonuclease, which will break the double-stranded DNA. What else do we need? Yes? Exactly. So we need the homologous repair template, so homologous sequence to our gene, and then in the middle of where we are going to make the break, just put GFP sequence. So, as you just said, we're going to design the small guide RNAs to target cleavage at a position in the gene that we want to tag. We're also going to need to design our homologous recombination repair template, which will have the homologous sequence flanking the GFP. And we'll need those two reagents plus our Cas9 endonuclease, put it into cells. The break, the cleavage that will happen because of that endonuclease will then ideally be repaired by our homologous repair template and insert GFP where we put the break. In this case, we put it at the very end of the open reading frame. Would we put it after the stop codon or before the stop codon? Before the stop codon, right? Otherwise, what will happen? Translation will stop. We will not get any GFP tagging of our protein. So we put it before the stop codon so that the protein is fused to GFP, and then we can see this endogenous protein. What cells is it expressed in? Where in the cell is it expressed in? Any questions about that methodology? Yes, Antonio? Sure.
So does that mean that you're going to define the
They are, so there will be a messenger RNA that will include all the codons for your open reading frame and then the codons for GFP. When that gets translated, you end up with a protein that has this open reading frame fused to GFP. For many proteins, they function just fine, having these, what we call a tag or a fusion. Some proteins are picky about it. We actually first tried to put GFP at the N-terminus or the beginning of the Argonaute protein, made it completely unfunctional. So you have to be careful about when you tag an endogenous gene, when you're changing its sequence, because some really care about like what their folding looks like, and it can ruin its activity. Other questions? Yes?
If our sgRNA is not like specific enough, will we have like really more off-target effects?
Yeah, so you want to be really careful about how you design your sgRNA. What method would you use to try and find a very specific sequence in your gene of interest? Before you even like do the experiment, so you're just designing, maybe you have 3 different sgRNAs that you might want to use. How would you choose and make sure that at least one of them is specific? Yes? Blast, exactly. Take your sequence, blast it. If it only shows up to match one sequence in the genome, then you are very likely to have good specificity. But we'll come back to that question also in a moment too. But yeah, your goal is to find a sequence that will only match one position in the genome. If you don't do that and there's multiple positions, you're going to get cleavage at multiple positions throughout the genome. And unless you intended to do that, that's usually a very bad thing to happen in an experiment. Any other questions on that? All right, I think this is our last clicker question. We'll see if I can get the clicker to come back up here. All right, so you want to use CRISPR to insert GFP coding sequence, as just shown here in green, right after the start codon of this gene X right here using this template. So which of the following templates would you add with your guide RNA and Cas9 in order to insert this GFP right after the start codon in this gene X? Ten more seconds. All right, final answers. All right, people are mostly liking C. No one voted for E. Thank you, there's no E. All right, who wants to lead us through this? All right, Cleo.
Just C, because like you can see like the first ATG up there.
Mm-hmm.
So the GFP sequence must be like a tree in there.
Yes.
After the ATG and before the CGA.
Yep. Yeah, so you only have to look at sequence right before where the GFP is going to be inserted, so it's going to have to match this. And then on the other side, you want the homologous sequence, so it will match that. You more clearly see it here. So when you design your cleavage, it will cut, open up, and then you want to have homologous sequence flanking what you want to insert. In this case, it was the GFP sequence that you wanted to insert. think most of you got that. Any questions about that or designing that? Yes, Nick.
What was the point of D, like, having it flip on the bottom of the start and stuff?
Don't choose this. Don't do something. Yeah, that's me. This was like, this would not make any sense, right? So if the GFP stop codon, this would be an example if you did not use directional cloning to make this homologous DNA sequence and it flipped in the wrong way. This is not going to encode GFP. So yeah, so D, I think not many people chose that. C is the only one that would put it in the correct position and have the correct homologous sequence flanking that GFP coding sequence. Any questions about that? All right, so that's how you would design your homologous template. And then the last thing, of course, you want to do is you want to sequence to validate the change and confirm that it was specific. What kind of sequencing would you do to first confirm, did I even change the gene the way I wanted it to be changed? You just want to look at one gene. Did I change it? Did I insert GFP? Yeah, you could use Sanger sequencing. It's the way we want to look at one gene. What if we, right, so Sanger sequencing will tell us the editing is precise, meaning that the gene we targeted got the edit that we wanted it to have. But sometimes we want to make sure that we didn't end up editing other things in the genome, that maybe our guide RNA was not as specific as we wanted, and maybe we got some changes elsewhere. What is one way we could test whether we have unintended edits elsewhere? Next generation sequencing, right. This is the way we can tell if our editing is specific, meaning the only change we got in our genome is at the position that we expected. So we would take our CRISPR-edited cells, do next generation sequencing. You can compare the sequence then to the human genome. Do you have any other changes? Sometimes it's good practice to take the same cell type that was not edited and also do the next generation sequencing and compare those two, because sometimes the reference genome can have some changes compared to the cells that you are using in culture. So this is really important because first you definitely want to know, has the intended edit been made? And sometimes you'll screen many cells to make sure, okay, this one has the edit. And then you'll choose a few of those to do the next generation sequencing and make sure that it is specific to the gene that you intended and that you don't have unintended edits elsewhere in the genome. And in fact, we know that unintended edits can happen in CRISPR experiments, depending on the cell type or what you're using. So it's very important to check for this type of specificity. Any questions about CRISPR? Anything else we talked about tonight? The last thing, I want to thank you all for being here. I really appreciate this class. You guys have been amazing. Every time I asked a question or said, please, somebody tell me what you're thinking for the CRISPR question or for the clicker question, somebody always was brave enough to volunteer an answer. Often it was wrong, but that's just as important. Not often, sometimes. But that's really important, right? Because that generated discussions and is helpful to other students who might have also been thinking the same way. It's helpful for me to get the feedback and be like, this point is not clear. And so I really appreciate all the effort you have put into coming here, being a part of this class. I know this class is a lot of work. Molecular biology is very challenging. Many of you probably feel like you've learned more than you ever thought was possible, and that is wonderful. I really, truly hope that what you have learned in this class is going to serve you well in all of your very bright futures. So you're ready for that. You are ready for the exams. Don't stress. You're all going to do a great job if you put the effort into it as you have been doing this class the whole quarter. So thank you very much. That's it.